So we're going to cover a couple things today. First is in genetic engineering. That's part of what you're going to be heading into. And then we're going to talk about what a PCR does. Then we'll go back and do the Piccolo and possibly do a review there. So let's go ahead and get started. Start this slideshow. So genetic engineering, this is what we're all about in biotechnology and what's going on. What is genetic engineering? It is manipulating the genes of the DNA sequence within a cell or organism to produce a desired result. So what we did with our last one with Piccolo is we manipulated Goose. We manipulated the E. coli gene so it would glow. And you can see some of us, it wasn't the whole thing. We didn't get the whole thing to glow. We were able to just manipulate a little bit. It helps in a change of the genetic makeup throughout an organism. We can make it deliberate, or we can remove it, which is a deletion. So deliberate addition is insertion, deletion is a removal, and we can mutate the DNA intentionally. Now, just by having kids and having posterity, your DNA gets mutated. The most commonly involved transfer includes one gene from an organism to another. So that is having kids. How it's used in medicine is gene therapy. And this is where it's going to be coming a lot into things that are on the rise here, which is gene therapy. Transforming a sick person's cells like sickle cell anemia or cystic fibrosis. I'm going to have to separate you. We use a viral vector. Now, this is sort of cool because I've got a sister-in-law right now that has melanoma. That's not cool. But they've tried to take it out surgically, and it's on her head. And so she's got a bald spot about this big on her head, but it keeps growing back. And so she's in a trial down at UU, and they've actually taken the hepatitis virus and attached to it the cancer drugs. And so I'm not sure. I'm thinking it's Hep A. But they're making it injections so that it goes right into the spot, and they're seeing that the tumor cells are actually shrinking, which is cool. So that's something they're doing right now with viral vectors, and they're making it specific. So in gene therapy, we basically take some bone marrow cells and remove them. The bone marrow is isolated and enriched to isolate the parent cell, and that's sort of what we did with the Piccolo. So if you actually grew Piccolo last time, we took it out and we're hopefully growing it more. And if you grow it more, then we're going to be able to grab more of it. We infect it with a virus, so like I said with the hepatitis, with the melanoma, they've actually put that in there so it will attach and attack the melanoma. So then they can fix by putting it back into the person or the baby boy. Okay? So some other examples, oil spills, they're now producing bacteria that will actually eat the oil spill. Rather than trying to clean it up, they'll actually eat the oil spill. We also have bacteria producing insulin for humans. Bacteria that produces BST, which helps control the production of milk. Bacteria that's help us to digest stuff, not only with the oil spills, but lactase, that people now that couldn't used to drink milk, now can drink milk. So human insulin, this is something that to me is important because I am diabetic. Yeah, I really am. You can actually produce that. The problem becomes, and we talked about cost, the problem becomes you can't just buy a vial and some needles because now companies are putting insulin into specific pens, and so you have to buy that, and it's not free anymore. So to do insulin itself, it's just isolated from a DNA sequence. They splice that insulin gene in to the DNA of E. coli bacteria. Does that sound familiar? We did the glow. Okay. And the bacteria then carry the insulin for the production of, or the DNA for production of insulin. And it passes it on to the next generation. And then it's separated and purified, and the bacteria are destroyed. And then you get a supply of insulin. So pretty easy. The BST comes from bovine somatotrophin. It's a hormone composed of a protein that is produced by cows in their pituitary gland that tells them that they need to produce milk because they've had a baby. So that's the hormone. And they're able to give them that hormone so that the dairy cows can keep producing milk, even though over time they would not do that. It also helps them to produce less fat and more milk, so that the milk coming off is more what everybody wants to drink now, which is 1 or 2% rather than whole. By splicing genetic material again into the E. coli bacteria, that's the one we've used the most, is the E. coli bacteria. Yes, ma'am.
Sorry, kind of back to the insulin thing. They, like, all they do is, like, they make insulin, right, with chemicals. They don't have to take it from people that have died and, like, have pancreas and stuff.
Right. We'll be back in lab in just a minute, so if you want to sit for a second, and then we'll go back. Okay? She wants to take some pictures.
Have they ever done that? Have they ever taken, like...
They used to use pig livers and cow livers to make insulin, but they couldn't keep up with the demands. So that's where they came up with this other way to do it.
Okay.
Okay, in the 1980s, we used microbes to eat oil spills. It's bioremediation technology. So when we have big oil spills, instead of trying to sop it all up, and then what do you do with all these sponges that are full of oil? The bacteria actually eat the oil. Okay, so genetic engineering is part of what we're doing in biotechnology. We're going to isolate some foreign DNA. That's the first thing we want to do. And then we want to insert that DNA into a bacteria plasma and see what it does. That's what we're inventing or looking at. We're looking at the transformation. That's what it's called. Okay? So when we go to looking at DNA, we isolate it. It involves finding the gene sequence you want. Yes?
Sorry, I have
No, you're good.
question. What's the difference between bacterial plasmid and human plasmid? Like,
So humans don't really have a plasmid.
okay.
Okay, because we have DNA, RNA, transfer RNA, mitochondrial RNA. We don't have a plasmid. Plasmids are what are in bacteria. So that's their DNA, but it's in a circle.
Okay.
Okay, so we can use that bacterial plasmid to make things we need.
Like replicate human DNA?
Yeah, or the insulin, that part.
Insulin?
Yeah.
Okay.
Yep. So we're going to cut out the chromosome that we need. It's called, we need a restriction enzyme. So the activity we're doing was sort of figuring out when things stop and go. And remember I told you that they're using that virus? This is what a virus basically looks like. And they want that DNA to be up in here so that when it attacks, it's sort of like a mosquito. This base plate, it actually injects whatever it has up here in the head. And so that's what they're using to actually help with controlling that melanoma, which is really cool. Way to think about some way to deliver it in your body. A restriction enzyme that we'll be using is TAC1. It is this. It cuts between the T and the C. Okay, T and the C and the T and the C. Okay, so you got a little snippet going on there. It leaves sticky ends, C and G. Okay. We've got some sticky end thing going here. So we end up with a sticky end. Remember this? We're doing your activity. You get a sticky end. And so when we anneal it, when we heat it up, it starts to go together and wants to form together. Then we cool it down and we heat it up and we cool it down. That's called annealing. Okay? So we would go through and we would cut and we would anneal and we would cut and we would anneal and keep going. Okay? So she doesn't have to wait very long. I'm going to stop right here. I'm not sure how to pause this, but we're going to pause it. And then we're going to come back and look at our lab. And hopefully we'll see some of our glow lab. And then she can take her pictures and we'll come back in here and finish. Does that sound good? Okay.
Yeah. On this worksheet, it says to look at
Gloves and glasses and aprons.
it says to look at the amino acid chart on page 54 in DNA science, the first course to answer 10 and 11. Where is that?
Should we just coat your aprons?
Coats, sorry. Coats.
Because that's all we have left. So
Just put notebook as well.
okay.
Okay, that's good. Okay, where did I put my glasses? All right, let's go. Let's go see if we got some glow.
I'm scared. I don't want to look. If it didn't glow, I'm gonna start crying. You should get a picture of that. I'm gonna be crying.
Still shaking. If you got something on the shaker, you also want to go grab it and see if it grew glow. Grew glow. Some of it should be glowing, yes. Well, it should be mixed in, so I'll pull through the whole thing. Like it's separated. We'll find out. Okay, gloves are mandatory today. We've got E. coli going. And we don't want to have bad preference there.
Oh yeah, that's for sure.
You don't want to get into that, I'll tell you.
I don't want to be sending it in here. I have no clearance.
Glasses, gloves, and then two regular headphones.
Pray to the heavens right now and grew.
Okay, we got all your stuff, and we're going to have lights to be low. Have something cool. Please allow Alex to know so she can take a good picture of it for the yearbook. I hope it did too. I'll grab it. What? I'm not. I saw it popping in the assembly, and I'm not doing that. Popping in the assembly. Bad answer.
There's none for dropping this back.
Oh, you're not dropping.
There's no other.
Yes, there is. Okay, you almost ready? Can you turn the lights off? Go ahead and try.
Hopefully somebody has extra.
Is this whole thing below?
So did we. I was like,
We use some of his.
got this whole stack.
Okay, so then we start day one. Now, it's okay, guys. It's okay. It's okay to go back to day one. I will just. No, we're not wasting time. Because what do we need to learn? We need to learn how to do this correctly, right?
It's not our fault.
You need to write this in the bottom of it.
Right. This needs to be
All
day
this
one.
whole writing.
Just to say that.
So we didn't get to close, we didn't get to grow, we got the first flow, we didn't get to crop yet. So guess what? Everybody just start day one. Okay, day one again.
Okay,
So,
day one.
day one. So what do we do? What's the,
Oh, we see.
I don't even remember what it's all about.
where's your paper?
Do you have the? Oh, we need the ice bath stuff.
Yeah, the ice bag stuff, yep.